Welcome to this free webinar on bioinformatics for infectious diseases. In today's webinar, we will speak about bioinformatics for infectious disease research and present use cases for bioinformatics analysis methods that are used in viral evolutionary studies, antiviral drug discovery, as well as the emergence of drug resistance in bacterial pathogens. We will also showcase some of the latest developments on the Bioinfo platform designed for virology studies and introduce the upcoming Omics Logic training program for those interested in effective utilization of bioinformatics for infectious diseases, immunology, and microbiology. As part of our upcoming training program for students and researchers with a focus on bioinformatics for infectious diseases, today's webinar will introduce you to the ways host-pathogen interaction can be studied using NGS data analysis. My name is Beepsha Biswas, and I am the Omics Logic Community Manager, responsible for daily interaction with thousands of users that are part of a bioinformatics community managed by Pine Biotech. Over the past couple of years, we have seen participants from all over the world join our training programs, take online coursework, and utilize our big data analysis tools in various research projects. Let me start by introducing our company and collaborators to share with you details about the upcoming program. What we will discuss today and throughout this program is a result of hard work by an amazing team led by company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support training and applications in medicine. Dr. Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data. Pardon me, Dr. Anita, you cannot see the screen? Is the screen visible?
No.
Yeah, it is visible to me.
Okay, can everybody see it?
Yeah, it's visible for me as well.
Okay.
Yes, I can see it as well.
Thank you so much for letting me know.
Yes, I can also see the screen.
So why can't I see?
Dr. Anita, if you're having a problem, Dr. Mohit, if you can just help Dr. Anita. I'm sorry, everybody. Okay, thank you so much for letting me know. So I'll continue with the presentation now. Okay. So as I mentioned, under Dr. Brodsky's leadership, the Tauber Research Center started collaboration in research teams at universities like the University of California, San Francisco, Institute Pasteur, Tulane University, and many others, where research labs are working on cutting-edge research projects studying viral and bacterial infections and designing novel drugs and vaccines to treat and prevent infectious diseases. And the rest of the team that you see here, there are a lot of team members who are bioinformaticians, computer scientists, and we will meet some of them today in this session. The goal of the Omics Logic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. And I would like to request everyone to kindly mute yourself as the presentation is going on so that every participant can hear the speakers and the speakers can also interact with the participants. Thank you so much. And before we move ahead, I would like to invite you to join our social media groups, which are there in LinkedIn and Facebook. And I will just take you to our website so that you can see these different pages and groups that we have. And for Francis, who is asking if you can record the session is already live in YouTube and you will be able to review the session even later on. So I'm posting the links to the groups. If you're already a member of the group, thank you for joining. And if you are not yet a member, you are welcome to come and join these groups. And secondly, I would like to ask all of you to come with me to edu.t bioinfo. So let me paste that link for you here in the chat box. So once you're here, please let me know. If you already have an account on edu.t bioinfo, we have a step-by-step guide on how to make the most out of your profile on edu.t bioinfo. And if you do not yet have an account, I would like to invite everyone to go ahead and create a free account on edu.t bioinfo. And during the course of today's session, as well as the rest of the upcoming program, you will see that why we are asking you to create this free account, because we will be sharing a lot of the free resources. So while we are discussing, you can go ahead and create a free account on edu.t-bio.info. So let me go back to the presentation. And I hope that you're able to utilize the profile resource to update your own profile, and that is going to be really helpful. So now let me introduce you to the program mentors. In the upcoming Omics Logic program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the application of data science to biological data sets. Let me introduce you to the main speakers of today's session. Dr. Anita Gansha is a senior research fellow at Noguchi Memorial Institute for Medical Research at the University of Ghana. Her area of work is in genomics of infectious diseases. She applies data analytics to explore pathogen genome data for signatures of the emergence and spread of drug-resistant pathogens, virulence pathogens in response to disease control interventions. Dr. Gansha has also been an active participant in our recent Omics Logic programs and is now a collaborator and mentor in the upcoming Bioinfomatrix for Infectious Disease program. Dr. Mohit Mazumdar is our Omics Logic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is Pine Biotech co-founder and CEO. In his current role, he has extensive experience working together with industry and academic partners that are applied multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Like why bioinformatics is an essential skill for those involved in infectious disease research as well as health policy, how bioinformatics is used in projects and what are some of the examples, and what are the benefits of this study for students, clinicians, and researchers that Ilya also will be covering in today's session. Dr. Harpreet Kaur is our Omics Logic trainer. She guides the participants in the hands-on sessions. And with project and data analysis during the program, her area of specialization is in genomics and machine learning. I will now like to invite Dr. Anita Gansha to talk about her interest in bioinformatics and the way OmicsLogic training has helped her leverage data analysis in her current malaria research. Dr. Anita, over to you.
Thank you, Bibsa. Let me share my screen.
There should be oh,
Yeah.
okay.
Can you see my
Okay.
screen now?
Yeah. Mhm.
Do you have the slide view itself? Right. So good afternoon to you all, and thank you for joining us. And good morning to those of us in the US. Thank you for joining us for the first in the series of bioinformatics training that Noguchi Memorial Institute for Medical Research, in collaboration with Pine Biotech, is organizing for infectious disease research. So this afternoon, I'm going to give you just a teaser. I know that it is after lunch, people are tired and all that. So I've decided to give you a cartoon version of why bioinformatics for infectious disease, public health, and research. My name, as was mentioned, is Anita Gansha, and I work at the Noguchi Memorial Institute for Medical Research. So my goal is just to show you how bioinformatics can be used to help us prevent us from dying from something that is very awful, maybe COVID. Yeah, so that is basically what I'm aiming to do this afternoon. So before I even start, I want to ask, what is bioinformatics? Is it for genomics analysis, for evolutionary studies? Is it for biological algorithm development? Is it for drug design? So basically, you can see how we are all scrambling to find our niche on the bioinformatics platform. Now, why do we need bioinformatics? As it stands, most of us, what we do is to collect a lot of data, and then we scratch the surface, do some analysis, and put the rest down and hope that one day we'll come back to it. I think this is why we need bioinformatics for our research activities. So bioinformatics is that discipline, the scientific discipline that merges engineering, computer science, chemistry, biochemistry, biology, statistics to make sense of data, ultimately for improving health. And so we see that the biologist is interested in the sequences, whether it is genomics, proteomics, transcriptomics data. And then the computer scientist is in charge of the algorithms. The statistician does the statistical analysis, and the chemist is also involved with the protein structures and how all these will come together for a good bioinformatics pipeline. Now, in public health, we are interested in health protection, surveillance, prevention, health assessment, health promotion, and emergence preparedness. So when there is any outbreak, we are interested in how we can fight this outbreak. So what are some of the questions that we ask in public health? We take samples from patients for diagnosis. We want to know what the patient has and how the patient can be treated. We study samples for surveillance. So what is circulating in the population and what does it mean for public health? And then we also take environmental samples to know what is circulating in our water bodies or water that we drink, food, and what this means to public health. Now, in case there's an outbreak, how do we investigate this outbreak? So for instance, we are all aware of the SARS-CoV infection. How do we investigate such an outbreak? These are some of the questions that are of public health importance to us. So we conduct surveillances to identify cases, people who are infected. And these surveillances can include molecular epidemiology, genotyping of the pathogens. So surveillance is identifying the various cases. Then we can also go further with the molecular epidemiology to identify potentially which pathogens are related. So we see some relations between the identified pathogens in the surveillance. Then the field epidemiologist is interested in tracking the transmission, how the infection is being transmitted. But that is not enough. These genotyping methods that I spoke about in the previous slide may not necessarily tell you the order or the direction of the transmission. And then the size of the clusters that you attain from the genotyping will depend on the type of method that you use. So the more detailed you go, the more information you get about the pathogen you are interested in. Now, we need a lot of epidemiological data to marry these two. And most of the time, we do not have sufficient epi information to understand even the genotypes that we have attained. Now, in summary, these are some of the roadblocks that we encounter. Diagnostics is very time consuming, and we end up not taking a lot of samples in the population to understand the actual dynamics of the infection. Now, coming up with the exact phenotype is very, very slow. It takes days, especially for diagnosis like TB. And then the surveillance that we currently do are very low resolution because we are using genotyping or methods that are low resolution and then relies a lot on epidemiological data. So the whole process is very complex. There are so many parallel steps and reporting at various stages. And so these roadblocks we need to surmount. Now, how do we surmount such a roadblock? This is where we need genomics. Genomics promises to help us surmount some of these roadblocks. Yes, I know it's still early days. Some parts of the world have made a lot of progress. We are trying in Africa to also apply genomics in our public health systems. So we are all aware of how the technology has advanced. We have various sequencing platforms. I don't even know the names of these ones, the MiSeq, the HiSeq, and the more where we think the future will be, the ion torrent platforms. We think that at the moment, we are thinking that we can use whole genome sequencing to help with rapid diagnosis. We believe that the genomes of these pathogens contain almost everything that we need for diagnosis, for phenotyping, and for surveillance. So genomics can be used for diagnostics. The second thing that we also believe is that genomics or whole genome sequencing can help us tailor therapy. So instead of going from picking a specimen and then doing various drug sensitivity tests for especially TB diagnosis, and then testing various drugs to come up with which drugs we think that the patient may respond to. We want to get to a point where we can sequence the person's genome and then align these sequences and have some kind of algorithms ready that will link your sequence types to the drugs that one would need. So basically personalizing therapy. We also think that we can use whole genome sequencing to track outbreaks. So we're following an individual in the population who is infected. We see how the next person is also infected and how it is spreading in the population. There are some changes in the pathogen. We can see that there is a new variant in the pathogen. However, at this stage, we are just tracking the infection. So we can go further to use this to manage the outbreaks, see whether the activities are directed at the right persons and at the right locations. But as you can see, this man here is asking, is that all? No, it's not finished. We've forgotten about within host genetic diversity. So this is where we start looking at how a pathogen in an individual is changing or branching into the B type or the C type. And basically, this is where the statisticians will bring their statistics on board. And then we can eventually develop a nice phylogenetic tree to look at how these pathogens are related and on a time scale. Now, furthermore, we can also merge a number of these phylogenetic trees based on social networks, let's say various groupings in a society, and come up with a more complex analysis like this. Then we can also marry the epidemiological data to get a final construction of a network of how the pathogen is interacting, the pathogens are interacting within individuals and also with transmission in a population. But it's still not done. There are also certain very minor variants within this population that we can further mine, further complexing the kind of analysis that we can do. Yeah, you may think that is all. No, there's still a lot more that we can do with whole genome sequencing or NGS. We can use it to understand the dynamics of infections. So basically, if you sequence and assemble your genomes, and then you build your phylogenetic tree, you can plot the prevalence of these subclones across space and time to understand how and why the epidemic is happening. So for public health, this is one interest. The next thing is also to use these sequencing data to discover completely brand new pathogens. So for instance, if you do a metagenomics analysis of a patient with an unknown disease, you can blast the reads against the database of known organisms and look at the set of reads that didn't match to any known organism, and then try and sequence in the lab and piece various regions together. And if it is worth calling a completely known pathogen, then you do so. Now, for something like SARS-CoV-2, we are comparing this novel pathogen with other genomes within the genus coronavirus grouping, and to identify which regions have changed by comparative genomics with a known coronavirus, and we established the new SARS-CoV-19. Now we can also use sequencing data to understand the drivers and evolution of within host and population level evolution of resistance. to these pathogens. Next, what else we can do is to track the date and time of a viral infection, not only viral infections. From our phylogenetic tree, we can pinpoint the actual time of origin of a new variant in the population. We can also use genomics data to do this therapeutic monitoring of drug resistance. So you sequence an individual or a patient, and then you scan this genome for mutations and determine which drugs would work for the person. If probably the person is already on a certain drug regimen, you can change it based on the genotypes that the individual has got. We can also use pathogen genomics to understand the population structure of these pathogens, to see how various certain pathogens are clustered or grouped and others are separated. So we see this substructuring within the population to understand which areas, or for instance, which countries have similar pathogens, and basically how the pathogens are structured in the various populations that we are interested in. Now, for drug resistance, we can also use pathogen genomics to predict resistance. There are many mutations in the genome. Some may be compensatory, but some may also lead to resistance. So you can look at it, which of the rare variants will predict a resistant phenotype. We can also use it for diagnosis. People are actually developing new diagnostic tools based on the sequences that we generate from pathogens. And then in epidemiology, we will use it to infer transmission of genomics data. It's not enough to just sequence your genome. You need to marry it with the epi information of your population to understand the transmission, the dynamics of the transmission, and how basically the pathogen is driving this transmission. Now, I can't stop without talking about malaria. That is my area of interest. So I'll do a few slides on malaria before I hand it over to Ilya. So for malaria control and elimination, basically we are targeting malaria elimination and ultimate eradication. Our goal is to come up with new tools that will be useful for diagnosis, for prevention and treatment, and for stopping transmission or prevent reintroduction of parasites in areas with zero prevalence. So as we are controlling the malaria parasites and the vectors, and we get to elimination, what we are expecting is very, very low transmission. And when we get to the zero prevalence, that will be eradication. But as we go on, transmission is reducing. So imagine that we will need very innovative ways of diagnosing the disease in, let's say, 1 in 200 or 1 in 1,000 people just getting malaria. How can you pick such a low infection in a few people, and how effectively we can prevent and treat the disease? And also, if there is a reintroduction of the parasite in a population where this parasite has already been eliminated, we can quickly localize this infection and then deal with it. Now, what do we have in our tool set at the moment? We have a number of vector control interventions, such as indoor residual spraying with insecticides, and then long-lasting bed nets to prevent, treated bed nets to prevent transmission. And then we have preventive therapies such as SP, sulfadoxine, pyrimethamine for pregnant women, for children, and then intermittent treatment of populations with low transmission. And then we have our diagnostic kits, microscopy, rapid diagnostic testing, and we have our current artemisinin-based combination therapy for treatment. And we've established quite a strong malaria surveillance system. And we have one vaccine that is at the pilot implementation stage. So it's gone through, this is the phase 4 pilot implementation. It's been approved and currently being tested in Ghana, Kenya, and is it Malawi? Yeah. They're thinking about how it can be rolled out successfully in our countries. So these are what we have in our tool set. But the challenge is the extensive genetic diversity in the parasite genome. So you see how diverse the parasite can look like. And for the malaria parasite, there are many levels. From the mosquito stage, it adapts in the mosquito through diversity, and then it moves into the liver stage of the individual. And there again, it diversifies its genome in order to be able to evade the liver stage, and then moves into the red blood cell. And again, it has to undergo extensive diversity. So this makes it very complex for any malaria genomic studies or malaria intervention. The idea of the parasite's extensive diversity is just to evade the host immune system and pressures such as the drug and the vaccines that we take. And unfortunately for us, the current anti-malarial therapy, the artemisinin-based combination therapy, is already showing signs of resistance. The parasite is resisting this drug. And we see this in the greater Mekong region around Cambodia, Thailand, and the rest. And recently in Africa, in Rwanda, resistance to the ACTs has also emerged. And we already have insecticide resistance also in the vectors, the mosquitoes. And there is a deletion in the genes that are used in developing our rapid diagnostic test. So what are we left with? Yeah, but the good news is that with all the advancement in technology, for malaria elimination, we have a number of tools. We are evolving from doing simple nucleotide or nucleic acid amplification techniques like PCR for genotyping, molecular barcoding. We are advancing to microarray technology, whole genome sequencing. We also have whole transcriptome sequences, and then that gene drive using techniques like the CRISPR-Cas gene editing. Then we also have a huge genetic or genomics database or repository that we can tap from. Some of these resources I have listed here. We have the human genome, so we can look at the human genome, the parasite genome, the mosquito genome is also available. We have Plasmodium, GenDB, the malaria gen PF3K genome. We have the mosquito 1000 genome, NCBI, GenBank, and VectorBase. These are some of the tools that have stored a huge database. Yes, you may not find enough metadata to go with it, but there is a lot that we can explore together with our own sequences that we have generated. And some of the questions that we can ask by using whole genome sequencing or any genotyping platform is to understand or get some insights into the parasite's biology, its ability to adapt and track these parasites as they respond to interventions like the drugs or vaccines, or even now the parasite genomic data is being used as proxy for vector control interventions to understand transmission. The reason being that as the vectors are being shrunk by the interventions like the indoor residual spraying or the bed nets, they shrink with the parasite population. And so these parasites remaining after these interventions have become proxy for estimating transmission after vector control interventions. We can also look at genetic mapping by linkage or genome-wide association studies. Genome-wide association studies have been used to look at regions of the genome that are under strong recent selection. And these have been seen for candidates for drug-resistant candidates. New drug-resistant candidates have been developed or detected using genome-wide scans for these signatures of recent selection. And then they can also be manipulated to look for causal variants. So if you're interested in phenotypes like severe malaria, are there any causal variants to severe malaria? We can also map the genome or transcriptome to look at differential expression patterns throughout the various life cycles of the stages of the life cycle to enable us identify new drug and vaccine targets. And this is an area that not much has been done. We also need preemptive surveillance of the African parasite population for genetic markers of emergence of drug resistance, because apart from just tracking the genotypes that have been, the genes or variants or mutations that have been associated with or have been identified as functional for ACT resistance, there are signs that we may have different mutations apart from what has been established in the greater Mekong region. So it's important to look for what is linking drug resistance in the greater Mekong region. I'm talking about the K13 gene here. There are new variants in the K13 that may explain or give us a different pattern for our resistance. And of course, we know that the K13 gene alone is not the gene responsible, may not be the only gene responsible for artemisinin resistance. So there is a need to look for other markers of drug resistance. And then we need new tools to develop drugs, vaccines, insecticides, and diagnostic tools. As I mentioned, there are deletions in the genes used for the rapid diagnostic tech. And these will require some in silico designs, but we also have to think about validating whatever signals we pick from the genomes or transcriptome or proteome with laboratory validations. So the long story is summarized here. I'm saying that you should take home the fact that if you have a clinical diagnosis of an infectious disease, you can look at the epidemiology or laboratory procedures. That is what we do. We do disease population surveillance, disease outbreak investigations. We take samples and isolate DNA, and we do traditional identification, look for drug resistance. But we can go further by using sequencing and bioinformatic tools to get more insights in the sequences that we generate. So we do our PCR amplification, we assemble our sequence, and then we do our pathogen identification. You map it onto reference genomes. You do your comparative genomics and phylogenetic analysis, clinical genotyping, but with this huge database, huge sequence database, we will get more insights that will lead to discovery of new drug targets, new vaccines, understand the biology behind our pathogens, and more insights into the emergence of resistance or how resistance, the mechanism underlying resistance in a huge area of immune responses that I'm not even touching And ultimately, this will inform public health for disease control and prevention, and then clinical medicine for our well-being. So I thank you for your time, and I'll hand over to Ilya to continue with the presentations. Thank you.
Thank you, Anita. If you could hold for just 1 second, there's a few questions that people want to know. So Abraham Paul is asking, thank you for this presentation, Dr. Anita. Please, what is your group doing to improve the malaria diagnosis? Are you working on alternative diagnosis, maybe cheaper, rapid, or more sensitive techniques for malaria? What is your malaria diagnostic focus in your group? Thank you.
Thank you very much, Abraham. Well, I personally am not currently working on diagnostic tools, but at Noguchi, Dr. Linda Amua, she is spearheading the National Malaria Control Program's surveillance of this HRP2 and 3 deletions in Ghana. And I know that ultimately she'll come up with new diagnostic tools. She's also exploring the genome. So that group is handling diagnostic, but definitely there is some work in exploring the genome. Having seen that the HRP2 gene or 3 gene, there are some deletions, and therefore we are not picking them, and they are not properly diagnosed in malaria.
And another question is, what is the status of Plasmodium population structure in Ghana? From Francis.
So the Plasmodium population is extensively diverse in Ghana. We don't have a complete Ghana Plasmodium population structure data, but we have looked at population structure in various regions in the country. We have 3 major ecological zones. So for the middle belt, for the coastal belt, and then the northern belt, we have looked at the population structure. Yes, in the northern zone, there are a few clusters, but ultimately it is very, very heterogeneous. So we think that the interventions are slowly breaking down the structure, and there's a lot of gene flow across all the sites in Ghana. And this is explaining the very extensive diversity and heterogeneous population structure we see.
Thank you. And the last question, I believe, what is the pattern between villages and cities? I will just mention for the rest of the questions, we will continue to speak about malaria. So after this last question from Francis, we'll come back to questions in a few slides after this.
So what is the pattern from village to cities? Well, so these are the things that we have to track. Unfortunately, we are not really tracking, but we expect a pattern because of a lot of migration. We expect to see a pattern. And again, as I said, the parasite population is very, very diverse. Unless we get a specific marker that we can use to track this particular pattern. We know that people are moving. Migration is very rife in Ghana, but the population is so diverse, and even though we have not specifically looked at patterns from village to cities. Yeah.
Okay, thank you very much, Dr. Gansa, and we'll come back to these important and interesting questions in just a few more minutes. So thank you again for sharing this important information. I think it was very fascinating to hear all of this. And I want to kind of turn your attention now to some more practical overview of this upcoming training that will start with a focus on how bioinformatics can be leveraged for infectious disease research. And specifically in the context of today's webinar, how can bioinformatics help us analyze host-vector pathogen interaction? So before I continue, I want to kind of connect some of the things that were set up until now. So when we joined, the first part was an introduction to Omics Logic, which is a program that is designed to facilitate bioinformatics training. And we also heard about Dr. Anita Gansa about how bioinformatics could be applied in so many different ways. And I think what's important for anyone who is starting to utilize bioinformatics is to have a complete roadmap for bioinformatics. And so what I will try to do in my portion of this presentation is to help connect some of these important and significant topics in infectious disease research with specific practical hands-on tools that anyone can use. And I want to try and make it as broad as possible to involve people that maybe have zero background in bioinformatics, as well as people that might already have some experience in bioinformatics, but would like to enrich their skills and make the most out of this upcoming opportunity for training. So a complete road for bioinformatics, in my opinion, has to do with finding data sets that might be able to be used for answering the types of questions that we've discussed, some practical skills on exploring and visualizing these data sets to guide our hypothesis development, to understand research methods that are being used in cutting-edge research publications so that we can implement similar methods to analysis of our own or similar data sets, and gaining insights and understanding that are validated and we are confident in. So really that is the objective. And in today's webinar, what we wanted to talk about is a little bit how bioinformatics could be leveraged in very practical ways, and then to talk about some examples that we will cover in the upcoming program for those that might be interested in some of these aspects. So as you know, the major difference between infectious diseases and non-infectious diseases is that infectious diseases in general are caused by pathogens and they spread. And so they're also called communicable diseases. But as Anita already pointed out, there are so many factors involved. And one of the primary factors beyond the study of the pathogen itself is how does that pathogen cause disease, or what is the process of pathogenesis? How do these pathogens replicate? How do they cause different toxins to be released and harm the host? And ultimately, how can we study the impacts of this disease progression at different stages and caused by different pathogens? Another important part of that is obviously how can the immune system be leveraged to stop, prevent, and protect people from these kinds of pathogens. And so in the context of vaccine design, what Dr. Anita already mentioned, we have a number of important interventions that have been designed maybe over 100 years ago already that have shown to reduce the rates of infection and protect the larger portion of the population from such infections. And these are not just the vaccines. In fact, some of the most basic things like use of chlorine in water, or for example, separation from different types of species have caused an immense reduction in the rates of infection. And then the vaccines have also revolutionized the way that people have dealt with infections over the past decade, or over the past, 100 years. And so today, when we're looking at how we arrived at a place when we are again dealing with a pandemic that is surpassing the rates of death from other non-communicable diseases, is something that requires us to rethink this strategy of prevention and antiviral intervention or anti-pathogen intervention measures. So when we think today where we are in our ability to understand how pathogens overcome the traditional methods, how new pathogens are causing important, dangerous outbreaks like we see in COVID-19, and how we all know that COVID-19 is just one of coronaviruses that have been around and have been causing mild disease in the past. So how can similar changes in pathogens can ultimately cause another outbreak, and how can we be prepared for that? And my proposal here is to say that as researchers, all of us play a major role in that preparedness. And a lot of what Dr. Gansa already mentioned, including the understanding of the basic mechanisms of replication, transmission, and pathogenesis, our understanding of how the virus causes disease in the host response, and our ability to leverage such data in a rapid way to quickly identify insights that could be practically translated to active measures. We are talking about essentially putting all of this data in context. So what's really amazing is the speed at which such data has been gaining or accelerating over the past maybe decade or two. In fact, today, we live in an era of exponential growth of structural data, genomic data, transcriptomic data, all kinds of high throughput data that ultimately together indicate how hosts interact with pathogens and how we can leverage the immune responses and the natural defenses of different organisms to really understand disease and be much more informed about the opportunities to be effective at intervention. And so what we want to do in this training program that we're going to describe in just a few more minutes about how this program is going to be organized, delivered, and how all of you can register, we would like to focus on a few different types of infectious disease data sets. And that includes viral genomics and evolution, immune response and biomarkers, and the microbiome and infection. And in fact, if you look at this kind of data, you will see that a lot of such data really contextualizes what we already know. So we see that from the time that the human genome has been sequenced, now there has been a parallel growth in our understanding of the human genome, as well as in the genomes of different viruses that have been studied in the context of the microbiome and in the context of what's called the virome, which is probably even more complex than the metagenome. And so when we look at the changes in the way that sequencing data is generated, when we look at the ways that different types of data can categorize different forms of life, or even how we can study different forms of infection that are caused by maybe non-living things like viruses, and try to place them in the categories that we have to deal with in the tree of life. So the key technology to all of these, as was already mentioned, is next generation sequencing. And one important part to start leveraging next generation sequencing effectively is a workflow to help transform next generation sequencing into structured data that could be readily analyzed by anyone. And so one of the things that we would like to introduce you today is a platform that can help you gain the most out of available data sets that you might have generated in the lab or that are publicly available, so that you can generate hypotheses from publicly available data sets or be more effective at extracting greater insights and more detailed and precise insights from the data that you generate. So what's out there in terms of publicly available next generation sequencing data? So first of all, we can see that in the past 2 decades, there has been an exponential growth of genomic data about pathogens in different public repositories. And that includes different types of repositories like the NCBI. And so you've probably heard of the recent kind of update to NCBI virus, where there are over 13,000 genomes. And if we're talking about malaria or other kinds of non-viral diseases, not NCBI virus, but NCBI itself has also huge collections of these genomes. But they're also specialized repositories for specific diseases. Not only that, today, as we all know, we hear about genomic data all the time. And so even the greater population, not researchers and not students of these topics, are now familiar with the relevance of identification of specific variants, the impact of variants on transmission and pathogenesis, and how critical this data is to our ability to understand and track these infectious diseases. Now, beyond that, we also have the host data. So for example, on the gene expression omnibus, on the NCBI repository that is dedicated to gene expression studies, there are over 80,000 publicly available data sets that speak about drug response, that show us how pathogenesis happens, and allow us to interrogate how different types of genomic variations from the host can have a difference on the way that disease is manifested in different hosts and how it is carried by different vectors. And more importantly, there are novel techniques that are emerging, like metagenomic and viral sequences, or even real-time sequences of outbreaks that allow us to understand the disease and contextualize it with what's happening around that. For example, how that relates to the different measures that the health ministry can implement and how that has to do with different types of interventions. So I think that we're really living in an era of big data that is defined by the different types of data that is defined by also different types of applications of data. So it's extremely significant that we all have the ability and the tools to effectively manage, analyze, and interpret such data efficiently. And so one important thing about this is that not only has the data become more and more affordable, as a result of the diversification of the different types of data generation, we can see also a variety of different formats. And so before I continue, our interest in this webinar is to present you with the upcoming program and tell you some of the topics that we will cover, but it is also to get to know you. And so before I continue, I obviously do not know many of you or the majority of you. And so I would like to propose a few questions so that we can ask and get the information about the current participants. So the first question that I have is, what is your current level of education? So if you could please look at these questions and put the response by putting, for example, 1B for undergraduate degree, or 1D for postgraduate. So the first question is, what is your current level of education? 1A, high school or no degree. 1B, undergraduate degree or bachelor's. 1C, graduate degree or master's. And D, postgraduate, PhD. And that also is for those that are researchers and faculty. Okay. Thank you very much. And by the way, we will use this information to further tailor our webinars and sessions in the future to provide you with more information relevant to you. Okay, excellent. Thank you. So I see a lot of 1Bs and 1Ds. All right, and 1Cs also some I see. Okay, great. Excellent. Thank you very much. And my second question is, this webinar is introducing you to an upcoming training program that is hosted by the Noguchi Memorial Institute for Biomedical Research. And so we are interested to know how the information that you've seen so far could be interesting for you. So the questions for the second question is, what do you expect from this program? 2A, learn about bioinformatics. 2B, get familiarized with infectious diseases. 2C, overview of bioinformatics and virology. 2D, apply bioinformatics to a research project. So what would you say characterizes your personal interest the most? Okay, excellent. So a lot of 2Ds, which is applying bioinformatics to a research project. And I assume that is because most of you are already studying infectious diseases, and hopefully out of this session, you would get a little bit more information of how bioinformatics could be applied to your research, and also what are the specifics, not just broad topics like we've discussed up until now, but what are the specific practical skills that you can gain from here. Okay, so I have another 2 questions. So thank you again for all of your answers here. Number 1, what is your primary interest in terms of your research? Because many of you answered that you're interested in research. So 3A, parasites like malaria, for example, 3B, viral infection like COVID, 3C, bacterial infection like tuberculosis, 3D, all of the above, or any infectious disease. So you're really disease agnostic. Okay, so here are some, so please, yes, make sure you answer with the right 3. Okay, 3D, 3C. Okay, so a variety of different interests. Thank you. The last question for today's session is, again, what is your primary interest in terms of what are you interested to apply bioinformatics to? And sorry, this should be 4. 4A, pathogen, 4B, host response, 4C, vector transmission, and 4D, host pathogen vector interaction. Okay, and I'll just give here a minute for anyone to finish answering these questions. All right, excellent. Thank you very much for your answers. We are going to analyze your answers to really inform our next series of webinars and the training sessions that we will have in the program. Let me briefly introduce you to some resources that we will leverage throughout this program. So as Anita already, as Bipsa already mentioned in the very beginning of this webinar, we have a training portal that all of you are invited to register in, and all of you are invited to start exploring, even creating a free account, because in the context of this upcoming training program, we will leverage a variety of resources related to this portal. So here, once you have registered, you will see a variety of courses that are designed around specific types of omics data that go from the very basics all the way to the more advanced bioinformatics methodology. And all of these resources could be used asynchronously to study on your own. However, for many people, having asynchronous resources like Coursera, edX, or the T-Bioinfo platform is not really enough. And so what we will try to do in this program is guide the participants from where you currently are, with or without any bioinformatics background, to understand the specific details of bioinformatics so that you can be independent in your research to ask questions of data and get results or answers to your questions. So in the context of this program, this is one important resource that you will have access to. And you can start looking over the courses here or some of the collections of courses and projects that you will find that take a specific data set and explain what that data set could be used in the context of a research question. Now let's talk about some specifics. So specifically, when we talk about bioinformatics, the journey of a bioinformatician starts with finding a data set. And so when we speak about genomic data, there are major 2 types of data that provide us with genomic information about the pathogen. We can look at FASTQ files that provide us with an in-depth look of what's in our sequencing data, or we can deal with FASTA files that can describe genomic variation that could later on be interpreted. So one of the applications of these is to understand the significance of mutations in terms of codon mapping and the impact of a particular mutation on the amino acid and even on the structure of a protein. As you probably are all familiar with, this could be done by leveraging multiple sequence alignment at the nucleotide or amino acid level and understand the relationship between sequences using what's called phylogenetic tree reconstruction. So one of the important parts of what we will go through in this program is take these different examples that we just looked at, Ebola virus, tuberculosis, malaria, and analyze those genomic sequences, both as consensus sequences in FASTA files, but also in raw next generation sequencing data, so that we can look at the in-depth mutation analysis that reveal haplotypes and different populations of host pathogens and how they interact. So for example, one of the things that we will do is we will look at geospatial analysis, where we can link specific sequences to their location and the time where they have been found. Another important use for such data is to find single nucleotide polymorphisms. And in the context of big data, it is a challenge because we will look at the impact of coverage on our ability to detect low frequency mutations and differentiate between low and high frequency mutations to study populations of pathogens, but also the emergence of dominant strains and how we can characterize those in large groups of samples. For example, when we talk about COVID-19, when the first sequence was put together, was assembled, that looked like this. You have a lot of different reads. Those reads have been mapped on some reference, and a consensus sequence has been provided. But when we talk about analysis of such data, there are many indicators that we can extract from such data that will allow us to ask very different questions about how this consensus sequence could be compared to other consensus sequences, or how can the raw FASTQ data can reveal different types of SNPs, different types of combinations of SNPs, and different types of information about their relationships. Now, in the context of such practical analysis, we will use our Bioinfo platform for big data. So let me show you what that looks like. So again, this is the educational portal, edu.t-bio.info. Now we can go to server.t-bio.info. So server t-bio.info. And so Bipsa, if you could please post the link and also a demo account. And so I want to introduce this platform in the context of an efficient tool that you can leverage in your omics data research. So here we will talk about how you can take FASTQ files or FASTA files. And by the way, thank you, Bipsa, for posting the link and the username and password. Anyone can log in with this username and password just to kind of explore some demo examples of what we will go through. And here, anyone, even without knowledge of bioinformatics and without access to high throughput computing resources with large RAM and large storage, can analyze publicly available data sets as well as private data sets that you have generated in your lab. So here you'll find many different sections for next generation sequencing data analysis, for mass spec, structural biology, as well as data integration and modeling, and especially a section that was designed around virology studies. So here you can see a number of different examples, for example, how you can build a phylogenetic tree. And this could be done using FASTQ files or assembled genomes in FASTA files. We will also look at how to analyze transcriptomic data. So for example, how we can build a whole pipeline to study patterns in our next generation sequencing data for transcriptomic data analysis. And this is a really simple tool that will eliminate the complexity of bioinformatics analysis to produce interesting results that you can gain insights from without any coding and without having to have your own high throughput computing resources. So here, for example, we are looking at an example of mapping a sample to a reference genome to find alignment of reads to the reference genome, and even look at how we can find variants in the sequence or understand what those reads represent. So these kinds of examples in the context of, again, viral infection, bacterial infection, and parasite infection, we will look at these kinds of studies as well as the host response. So what are the different sections that we will cover? We'll cover phylogenetic analysis, gene expression analysis, alignment with multiple sequence alignment or alignment of reads to reveal SNPs, variant calling, and even structural biology, how we can map those variants onto structures of proteins, understand the impact of mutations on the function of a particular protein. So as I mentioned, this platform will help you build the logic of analysis, combining multiple scripts into what's called bioinformatics pipelines, understand the different inputs and outputs in terms of the logic or the sequence of how they are connected together, understanding each method that are explained as you build your pipeline, different types of inputs and outputs for analysis, including this example of transcriptomic data analysis. You will see how such a guided process with the help of our trainers will help you establish a logic behind these workflows and make reproducible research. And importantly, you will find access to a variety of different sections that are all built around a similar interface. So using this platform and a number of other resources, we will talk about sequence variation, evolutionary relationships, relationships between function and structure, and host response, either as a vector or as a host. And so that way you can start contextualizing some of these questions that we discussed in this webinar in the context of available data sets that we will also introduce you to, and in the context of how to transform your questions into a specific transcript, in a specific analysis approach. So to conclude, I wanted to open up now a question slide. So if you had any questions about this program, you can post that here. You can also post questions about some of the topics that we discussed related to analysis. And to help guide us in this discussion, I also have here a number of questions that you can think about so that we can have an effective discussion. So the first one is which university or department you are currently in. Then how often do you deal with next generation sequencing data in your lab or in your research or in your studies? And typically, is it generated by you, or have you or do you currently leverage publicly available data sets, or maybe both? 3, what is your current training experience with bioinformatics? Sorry, this should be 3. And this should be 4. And how needed is additional training in bioinformatics for you or for your department? So these are some questions to help guide this discussion. You can definitely put your answers into the chat box. And if you are interested in kind of unmuting yourself and asking a question or answering one of these questions publicly, please feel free to do that as well. Okay, so I see that Rhodes University, James is answering that he's an intern at the Noguchi Immunology Department. Manav Rachna Dental College. Okay, and I'm assuming that the majority here are from the Noguchi okay, so I'm not so familiar with the abbreviations like KNUST and KCCR. All right, thank you. Humboldt University, PhD student, University of Health and Allied Sciences. Excellent, thank you very much. So we hope that these free sessions like today will be of interest and beneficial to you to not only learn about bioinformatics, but also gain some practical exposure to bioinformatics methods of analysis. We have included a variety of different free resources that most of you will have access to, but also in just a few moments, many of you will be introduced to our upcoming training program, which is a paid program. And some of those paid programs will also include some free webinars. So we'll go over that schedule in just a few minutes. So as you stay, you'll see more information on that. Okay, Sweden, Ghana Medical Center, Biochemical Engineering, Indian Institute of Technology, BHU, undergraduate and junior. Okay, thank you. Victor is saying has not yet gained access to next generation sequencing data. All right, thank you very much. Suez Canal University, Egypt. Okay, thank you, Dr. Mena Aboil-Care. Okay, thank you. PhD student biotechnology from Ben Gurion University in Israel. All right, great to see you here, Abraham. NMIMR, University of Ghana. Thank you, Dido. Okay. Lewis is saying that he's aware of the data sets available and hope to use them. And Victor is saying that he has basic experience on bioinformatics beginner. University of the Western Cape, South Africa. All right, excellent. So again, thank you all for your answers. We will just keep this for one more minute. So please answer some of your questions. And again, we will also be interacting with you. So as you're going through these questions, I also want to mention that one of the first courses that we invite you to complete in the context of bioinformatics training is a free Introduction to Bioinformatics Asynchronous course that is on the edu.tbioinfo website. And on this website, you will find that the first lesson is actually a survey. And so we're really looking forward to learning more about you, about your background, about your interests, hoping that we can leverage the resources that we already have to help you get more hands-on experience. Okay, so thank you again, all of you, for the answers. I think that an important part of what we have discussed today is not only the training and the lessons and the education, but I think that in many cases, we are interested in taking this further and applying bioinformatics tools to a particular project. And so to speak about projects, what projects are and how such training is going to help you develop a project of your own, I am going to pass the presentation over to Dr. Mohit Mazumdar, who is our project coordinator and business development person. So Mohit, I'll pass it on to you.
Thank you, Ilya, and thank you to other presenters, Anita and Deepsha. Let me share my screen. All right. Hello, everyone. A very good evening to all the participants. My name is Mohit Mazumdar, and in my current role, I'm collaboratively working with universities and research institutes and labs to provide support for bioinformatics research and training. I'm really excited to be a part of this OmicsLogic Infectious Disease Research program as a project mentor, and to be able to work on translational research with a diverse group of students, faculty with diverse expertise. So my PhD was focused on using bioinformatics and data science techniques to predict and identify biological patterns. And with this experience of working on several published research projects as a project mentor, I will help the program participants with analysis steps and understanding the sequence data and the pattern within, and further use structural biology tools and computational modeling of biological systems to develop a deeper understanding of the system biology. So, as you know, and as we have been discussing so far, bioinformatics and application of NGS and clinical data are increasingly contributing in both academia and pharmaceutical industry research and development. And this is quite evident from the rapid data growth generation that is happening right now. So our understanding of infectious disease caused by bacterial pathogens such as Mycobacterium tuberculosis and parasites such as Plasmodium falciparum, as well as viruses such as Ebola and COVID-19. So today, I will introduce you to several examples of how this can be done successfully by researchers who are studying viral infections such as Ebola virus outbreak in West Africa, COVID-19 pandemic, as well as microbial pathogen that we discussed, tuberculosis, and the single cell parasite, which is malaria. So we will briefly touch on viral genomics and evolution, gene expression patterns, transcriptomics that was discussed, detection of significant genomic variants, as well as look into the microbiome and the virome communities in a host. That will help us to find those important associations with disease susceptibility and those outcomes. So this kind of investigation is going to be really fascinating because we will be kind of focused on, as this has been discussed, about the disease outbreaks and pathogenesis, host pathogen genomic variations, and then host immune invasion mechanisms. So these are really exciting and very important topics that we will be talking about, including the identification of biomarkers or vaccine targets. So in the context of this training program, many of the projects we have been developing and will be used as an example. So mutation profiling of Ebola virus outbreak, as I mentioned, and then origin of and pathogenesis of novel coronavirus, as well as novel techniques on low frequency mutation studies, including SERC sequencing in poliovirus. So as you see that we will be using amino acid and three-dimensional structure and NGS data to be able to understand the research question that has been posed. So this research program that we will be doing and that we have been discussing and we will be discussing in the next few slides will include scheduled and structured training. And the purpose of doing this is this training should lead to a research outcome. So in this program, we will combine online sessions, guided study material. So we will have online sessions like the one that we are having right now. And then we have these courses and hands-on guides and tutorials. So we will use those. And then we will be using practical assignments for this immersive experience that has been proven effective in our well-respected Omics Logic programs. So in order to make the most of this training, we will be helping and guiding you throughout this training. So with this, let me invite our Omics Logic trainer, Dr. Harpreet Kaur. So Dr. Harpreet Kaur is one of the trainers that will help facilitate many of the sessions, and her areas of specialization is in NGS data, genomics, transcriptomics, and application of machine learning. So with this, I will give it over to you, Dr. Kaur.
Thank you, Dr. Mohit. Let me share my screen. So, hello everyone. My name is Harpreet Kaur and I have a PhD in Bioinformatics. During my doctoral research, my focus is on biomarker discovery for the diagnosis and the prognosis of the liver cancer. So what inspired me to take liver cancer as my doctoral research topic is the high mortality rate of this malignancy and lack of the precise biomarker that help to diagnose liver cancer at an early stage. So liver cancer is considered as sixth major cancer in terms of incidence and fourth in terms of mortality. So then what are the major issues that are associated with the liver cancer include its inaccurate diagnosis, and then its late diagnosis, specifically in the later stage. And then another reason that results for the poor prognosis of the patients is the tumor reoccurrence. So keeping these concerns in mind, we have tried to address these issues using various bioinformatics approaches. So in the first part, we have extracted the nearly 4,000 samples from the public repository that are associated with the hepatocellular carcinoma. And by employing different bioinformatics techniques, we have identified 3 gene-based biomarkers that can include CLAC1V, FCN3, and PRC1. In the next part, we focused on the second primary malignancy of the liver cancer, that is cholangiocarcinoma. For this purpose, we have retrieved nearly 900 samples from the one of major public repository, that is GEO. And then we have elucidated 3 CCS specific signature, which include LAMP-C2, ITZ, and C2. Employing various machine learning techniques like random forest, logistic regression, SVM, etc. And in next part, to identify liver cancer at an early stage, we have explored transcriptomics as well as the methylation data of the nearly 350 SCC patients. And then employing various statistical and bioinformatics techniques that include random forest, then various feature selection technique. Eventually, we have identified 51 signatures that include 30 RNA transcript and 21 methylation CpG sites that can predict liver cancer at an early stage. So keeping overall in conclusion from this my doctor research, eventually we have identified 3 gene-based biomarker to predict SCC and then to predict CCA. And then we have identified 51 signature to predict liver cancer at an early stage. So in my current role, I'm OmicsLogic Bioinformatics trainer. So with continuation to my research, here we are designing different research projects based on the public data sets. So for example, in the first project is already Dr. Mohit has shown you, that is based on the reason and the pathogenesis of novel coronavirus, that is SARS-CoV-2. So in this project, we will explore the NGS data to understand its origin and how much it related with already existing human coronavirus strains. And here in this project, further we will try to understand what are important components of the viruses through which they enter into the human cell. Subsequently, we also look into how it is different from other coronavirus strains in terms of mutations. So in the later part of this project, we also try to identify important signature that can significantly different under different therapeutic treatment and untreated conditions. Besides, we have also developed a project on another pandemic, that is Ebola. So in this project, we used next generation sequencing data to monitor the pandemic. Here we employed various bioinformatics approaches to analyze genomic sequences, identify particular viruses in the patient sample through the identification of significant variants, determine the relationship between viruses, and identify shared origin of viral genomes. And eventually we will try to understand to identify a connection between genomic variation and disease phenotypes. So during this upcoming program, many of you can explore these projects that are specifically based on the infectious disease. And I'm looking forward to become your project mentor during this program. Now I would like to invite Vipsa to talk about registration process. So over to you, Vipsa.
Thank you so much, Dr. Harpreet. I'll be quickly sharing my screen over here, and let me take you through the next steps, as we know that a lot of you have questions about how to register for the program, what is the schedule. So we will be going over them very quickly. In this upcoming 3-month training program, we will discuss in depth about bioinformatics for infectious diseases, go over an in-depth training in genomics, transcriptomics, as well as do a research project that already Dr. Mohit and Dr. Harpreet has been showing. We have designed this program to introduce the data, resources, and tools that you can use to apply bioinformatics to publicly available data on infectious diseases. In this program, we will understand viral diversity and its role in epidemic infectious diseases that keep re-emerging, including zoonotic transmission, transmission between humans, and the process of viral and bacterial disease development. We'll learn about bioinformatics analysis approaches to study viral genome and understand the relationships between viral strains, haplotypes, find differences in sequence data, and see the implications for drug and vaccine design. You will gain hands-on experience by analyzing curated data sets from public domain collections, guided by experts with bioinformatic experience and knowledge about virology. Here is an example of a project that Dr. Gansha was part of, along with her collaborator, Dr. Emeka, where they used transcriptomic analysis of SARS-CoV-2. So this project again is available on our YouTube channel, and I will be sharing the link with everyone after this program. If you're already registered for the program and have registered for this webinar, you will receive an email with the details to all of these projects and learn more about how this entire process works out. Now moving on. So if you're already a member of Noguchi Memorial, then you have to reach out to Dr. Anita, and you will also receive an email for us regarding the rest of the registration process. For those of you who are not part of Noguchi and are still interested in attending this 3-month program, so this is not a 1-month program, this is a 3-month program, and we will be dealing with all of these topics in greater details. And here you can see the breakdown of the different pricing on our screen. So I will just keep it on for a few more minutes. And there are also scholarships available for participants who are not able to afford the program prices. So if a participant indicates a high level of motivation and also provides us with a rationale to seriously consider this request, our team will reach out to you, and you will have to note down your references. And together we will reach out to you, talk to your references, and see whether you are fit for the scholarship program. And then upon approval, you will receive scholarship to the program. And the scholarships we offer are designed to help motivated, talented, and promising students to benefit from special training and research opportunities. The criteria for approval includes a reasonable need, indication of extra effort that the participant is willing to put into the program, and time commitment needed to go beyond the regular training. So to be able to get access to the whole program, a participant who is under a scholarship would need to keep a record of the topics covered in each of the sessions in what we call a virtual lab journal. And you would be required to give us a review of each of the sessions that you're going through. Make that extra effort so that we know that you're taking this seriously. And we will also be sharing with you what this scholarship looks like. And now, this is the first course that I had spoken about earlier, Ilya had also mentioned, which is the Introduction to Bioinformatics. So if you have not yet taken this course, you can go to edu.t-bio.info, and under courses, you will find this free course. And this free course also comes with a certificate. So you can get started with it right away if you have not yet done any of the courses. And you can see that there are a lot of reviews by other participants who have taken this course before you. And all of the activity that you do in the course of this program is being tracked to help you understand your progress and evaluate your effort at the end of the program to issue and for you to get a certificate and for us to issue that certificate to you. We found that the most successful participants are able to learn new methods, develop an independent project in the course of these Omics Logic program. Now, let me take you to the website and show you how the registration process works so that it is clear for all of you. Okay, Naim, I'm going to be posting the link, direct link to Introduction to Bioinformatics right now in the chat box. So for this program, many of you have already signed up and we have your details. So we will reach out to you regarding the next step. So please look out for emails from marketing@pine.bio, where I would be sharing with you details about the program, any free resources, details about upcoming free sessions as well. So some of these details may be useful to you. There would be emails which may not be useful for you because they may relate to any other topic that you may not be interested in. So we keep on sharing all of these resources from time to time. So you can choose to attend a session or not attend a session. So moving on, how to register for the program. If you have not yet registered for the program and are interested in attending this 3-month program, please fill out the form at the bottom of this page. So you see there is a pre-registration form right here. And once you fill out that pre-registration form, you will receive details about the program. And especially if you are someone who is, again, not from Noguchi, then the next step that you have to do is go to this page. This is the Infectious Disease checkout page. So I'm posting the link again here. Sorry about this. I'll just go back. Okay, I got the link. So in this page, you will find that the program starts with a hands-on workshop on January 29th. And it goes on till March 8th, where we will have a group project. So this is a program which starts with very basic understanding and builds up with the help of asynchronous courses, live sessions like we are doing today, where you can directly interact with a mentor, ask your questions, get them answered in a session, have your doubts cleared, as well as do hands-on analysis with the T-Bioinfo platform or any of our other platforms, as well as other tools, using other tools like R and Python, along with your mentor. So this is all going to be part of this 3-month training program. So please register yourself by getting started here. You will have all of these different options. The first step, if you have not yet registered again, is to go and pre-register yourself here. And then check out using any of these links. For those in need of a scholarship, we have another page where you can actually talk about your scholarship. Okay, I'll post the link again. I'm sorry about this. Yeah, so the scholarship link is now in the chat box. So if you're in need of a scholarship, please fill out the form at the bottom of this page. And please make sure that you will have to upload your CV and also mention your references. And we would like to have a meeting with you as well as your reference to understand where you are coming from and your actual need to get this scholarship and attend this program. So it may be a long drawn out process. So if you want to get started, please start applying for the program right now. And once you are enrolled in the program, you will get access to different resources like the course that you see here. So once you do each of these courses, each course carries a certificate on their own, as well as there would be a program certificate right at the end of the program. Okay, so if you have any questions, again, regarding the resources, please feel free to reach out to me, marketing@pine.bio, for any questions regarding access to the program, resources, courses, projects, anything. And I would be happy to answer all of these questions for you. If you are in Noguchi, you can reach out to Dr. Anita and Dr. Bright Adu, and they will be able to help you with the next steps of the program. And moving on, once you finish the courses, you're doing the online sessions during this 3-month period. You will also come across a lot of these projects which would be available to you, and some of which Dr. Harpreet has already shown you, like the COVID-19 project, the Ebola project, and you would be able to take advantage of these projects and have that hands-on experience on how to work on your own project and complete an own project, as well as conceive your own project and complete it, like Dr. Anita was talking about the malaria project, as well as her project with Dr. Chukbuka. So you would be able to do an independent research project in the next 3 months. So we are starting on January 29th. We do not have a lot of days. So if you're looking forward to register, kindly go ahead and register for the program. And now we are open to taking in questions. So if you have any question, please post them down in the chat box, or if you want to unmute yourself and ask any question, we are ready to take in any questions from your end. So please feel free to ask us your questions. And you can also continue to forward us your questions to marketing@pine.bio. And I would like to again invite you to take a look at our social media pages. So please go ahead and join our groups, as well as our group in LinkedIn. And Dr. Anita, do you want to add anything right now for the participants before we conclude today's session?
Thank you, Bipsha. I think that the participants, as has been said, if you are interested, you can register. But those at Noguchi, some are being fully supported and they know themselves, and the others would have to register. And you can get more information from Dr. Bright Edu of the immunology department as well. So you can reach out to Dr. Edu. Yeah.
Dr. Anita, can I share your email as well in the chat box in case someone wants to reach out? And you can also...
Yes, please.
Yeah, okay. So I will also post Dr. Gansha's link. Yeah, Vanshika, you can go ahead and ask questions. And for Francis, yes, we do have an intensive internship at Pine Bio for 3 months or 6 months period. So Francis, if you can share your email ID, I can reach out to you. You can send it to me in the chat box, and still I would like to take everyone one more time and show. So here is how you can register for the program, which is the research fellowship program, which we have. And as part of the research fellowship program, what we do is go through projects like you are seeing right now, and we have been taking some examples. So if you go through this link again, you will see the project examples from our previous participants who have been part of programs like this. So they come from backgrounds like neuroscience, they come from backgrounds like oncology, virology, and many more. So you will be able to see all of that project example here, developed as part of this research fellowship program. And Pradumesh, the recording, so the video is already live in YouTube and it does not get deleted. So technically the recording is available. So as you see, these are some of the projects that were developed as part of the program. And you can go through this link and let us know if you're interested in doing the research project. However, the training itself, the Bioinformatics for Infectious Disease program itself is a long 2-month program, which comes with first training and then the opportunity for you to develop your own project related to infectious disease research. In case you're interested in researching on any other topic which is not related to infectious diseases, definitely you can go for the research fellowship program. Are there any questions that you would like us to answer? Thank you so much, Francis. I will be reaching out to you. Thank you for sharing your email ID. So if you do not have any more questions, I would like to thank all of our speakers today for sharing their insights and for such an exciting session, and for all our participants for attending today's session, for asking your questions and for being part of this. So we hope to see you in the upcoming program, and thank you again for attending today.
Bipsa, before we go, is there another free webinar before the 29th?
Yes, on January 25th, I suppose.
Yeah, okay, right.
Yeah, so we'll be sending out those details via email to everyone, so that if you're already already registered via this link, you will get all the details. And if you're not registered already, to just get program updates related to bioinformatics for infectious disease, again, please go ahead and just register here. And we will be sending you all the updates once we have your details. So thank you again.
Thank you.
Naim, you can completely fill up the scholarship form and we will be reaching out to you. So thank you again. For any other questions, please reach out to marketing@pine.bio. And I would end today's session. Thank you, everyone. Have a nice week ahead.